welcome everyone. Our first speaker is Dr. Chittaranjan. Chitta is actually a professor of biochemistry in the Department of Chemistry at Purdue University. And he's actually well known in the field because of his early contribution to the studies of deubiquitinases. And I myself actually have borrowed very valuable critical reagents from him to study some DUBs. But recently he also actually is interested in how pathogens hijack host E3 ligases or DUBs to achieve to help with their life cycle. And we actually very fortunate to have him here today because as you remember, Lan said when Lan was a postdoc in Nikola Pavlich's lab, she studied the first E3 ligase, E6AP, and that was a milestone. Back then, it was an opening study, but not a complete study, okay? So I would say the chapter actually now is more or less closed by Chitta, who's gonna show us literally how this viral protein hijack the first E3 ligase ever identified in the studies of ubiquitination pathway and the structural mechanism of this system, so we can learn from virus to come up with therapeutics. All right, welcome, Chitta.
Okay, thank you for this opportunity. First of all, I must, like Ning mentioned, this is a brand new project which is started by a student who is actually now in Ning's department with David Baker, University of Washington, trying to do a protein-based design for PROTACs, molecular glue. But so I'm just going to present in this the work that he has done. But I basically want to start off with, I'll go to the viral system, but I want to start off with the general, the study that we do in order to understand the structure and mechanism. And as Ning mentioned, we are standing here on the shoulder of giants, and she's sitting there. And our work was inspired by her first structure. And sometimes I look back at the structure. This is 2000 or 1999. And I mean, you can say it was not complete, but if you look at and project it back there, it was a monumental work, okay? And so very inspiring. Okay, so I want to give you, because you had lunch, you might have forgotten about ubiquitin system. So this is a quick summary about how ubiquitination works. Here is a protein substrate that is being polyubiquitinated, and as a result is being targeted for degradation by proteasome. And this process is catalyzed by three separate enzymes, coordinated actions of E1, E2, and E3 enzyme, and requires ATP as a nucleotide cofactor. And so since we're going to talk about HECT type of E3 ligase, I want to dive a little bit deeper into this E1, E2, E3 transfer mechanism, especially to focus on or point out the distinct feature of HECT type of ligase as opposed to the dominant type of ligase that molecular glue and the PROTAC people are interested in targeting, okay? So the first in this cascade of ubiquitin transfer, we have the E1 enzyme activates ubiquitin through acyl adenylation, and then the activated ubiquitin is now installed on the catalytic cysteine of E1, and then E1 transfers into the catalytic cysteine of the E2 enzyme. Then E2 can bind to either a free E3 enzyme or substrate-bound E3 enzyme. Then in case of HECT E3 ligases, that ubiquitin is first transferred onto a catalytic cysteine of the E3 ligase, and before the ligase transfer into a lysine residue of the substrate. And then the result is the polyubiquitination, which I'm not showing you here, of the substrate here. So this is the basic mechanism of HECT type of E3 ligase, how they engage in this E2, E3 transfer cascade. Compare that to, for example, a ring type of E3 ligases, which, as I mentioned, most famous or most favored ligase systems for PROTAC and molecular glue systems is basically the ubiquitin from the E2 is directly transferred onto lysine of the substrate, with the E3 serving as sort of like an adapter that brings the two entities together. So when Kenny, my student, when he joined his PhD thesis program, he started asking this question, can you, we actually had speakers Craig Cruz came to the department. He presented his PROTAC idea. We were all excited about at that time. I didn't know that you guys are excited about molecular glue, but I hope I show you something in that direction that's still relevant to molecular glue. The virus protein that I'm going to talk about is actually, now that I listen to Ning's argument, is a natural molecular glue. Okay, we'll see how that holds up. But so he was interested in asking this question: can we target HECT type of ethyl ligase for PROTAC development? Okay, and so the approach we took was, many of you know that this is done to a lot of genetic siRNA, CRISPR-Cas9 kind of screening approach to find the ethyl ligase, an appropriate ethyl ligase or its ligand. So we thought we'd take a classical biochemistry approach where we'll set up a high throughput assay and search for ligands to HTS screen. So in the PROTAC modality, Michelle Pagano mentioned very well, nicely presented what is a PROTAC. I'm just going to quickly show you my version of it. You have, you want to bring a target protein of interest to proximity of this E3 ligase. And so you want to use a heterobifunctional molecule that has a target binding entity connected to the E3 recruiter. And such as this, he didn't show the structure of a PROTAC, so I'm showing that here. So here is this JQ1 ligand that binds to the BRD4 target protein linked to this thalidomide derivative that binds to cereblon called for complex. And so once you have that in your cells, now everything is there in the cell, all machinery. And so now your target protein is brought into proximity of the ethyl ligase, and through proximity-induced ubiquitination, you end up ubiquitinating that, and subsequently its degradation at the proteasome. So if you look at the literature, I think many of you are aware that the design of PROTACs has so far relied on a rather limited number of well-characterized E3 ligases, MDM2, VHL, cereblon, okay? And there are only a handful of those, and they're basically mostly cytosolic or nuclear. And Ning mentioned, and others might have pointed out, that people in the field are looking for tissue distribution, even different locations inside the cell. So you ask whether we can target membrane-bound ethyl ligases that would perhaps open up new avenues for therapeutics. So the ligase we considered, or we took up, was this NED4L. The yeast version is called RSB5, which is a membrane-bound ethyl ligase because it has this C2 domain that binds to targets to the membrane, and then has this WW domains which interact with this proline X, could be any amino acid, tyrosine, or PY motif in its substrate proteins. And most of the substrate proteins happen to be transmembrane proteins, receptors or membrane transporters, and in the cytosolic segment, they have at least one of these PY motif. And once the recognition of that substrate happens at the level of PY interacting with this tandem repeats of WW domain, then it is brought to the catalytic subunit, catalytic domain, where it's ubiquitinated, and then that has a proteasomal degradation, other biochemical fate. So the potential impact we could have is, we could think of a lot of receptor tyrosine kinases, and they have been regulated, dysregulated in cancer, and there could be other types of nuclear receptors, other membrane-associated proteins that would like to target for destruction, and the ligands already existing for those target proteins. So we thought, why not focus on finding a good NED4L recruiting element that would eventually allow us to expand this PROTAC modality. So this is the structure of RSP5, one of the WW domains. I think this is an NMR structure, but there's a structure from Brenda Scholdman's group, crystal structure of a different substrate peptide. But here you can see that from this protein SNA3, the PY motif is now, in this case, is complexed, and its NMR structure was solved. So essentially, in this system, what is understood so far is that one needs to first have this recognition of the PY peptide in the substrate at the WW domain before it's transferred, sort of the substrate is ubiquitinated at the HECT domain. So this may be debated, but there's this idea of the sequential substrate recognition. So we thought if we find a ligand that binds over here, the ligand will displace this, and then we will end up, if we have a good assay, we'll end up finding ligands that bind to the WW domain. And those ligands can be used in PROTAC. So the assay we designed was is shown here. You have basically, you have this NED4L, which is WW domain, not shown. And so we took a peptide from one of its known substrates and attached a fluorescent group onto the peptide. Then we argued if the peptide is recognized by the WW domain and is ubiquitinated, and then as the mass of the peptide increases due to polyubiquitination, we'll see an increase in fluorescence polarization. And that's in absence of any competing ligand. And if you have a competing ligand, if you imagine if you do this in presence of a high throughput screen format, if you have a ligand that binds to the WW domain or the catalytic domain, so basically it's going to displace the peptide. There is going to be no ubiquitination or less ubiquitination, so you'll see a much reduced fluorescence polarization. So the fluorescence polarization will be a readout of a binder to WW domain. So how does this assay work? So it basically here is how it works. We can show that in sort of a drop-off setup, if you take out any component of ubiquitin system, you essentially get no fluorescence polarization. But when you have all the required components, including E1, E2, the ligase, and the peptide, you'll see a nice time-dependent increase of the fluorescence polarization, which gives you a real-time readout of the ubiquitination of the peptide. And you can also see that on a gel, so that as this fluorescent ladder corresponding to polyubiquitination of the peptide. And if you were to add a deubiquitinase, a DUB, at some point during this real-time assay, it returns the signal back to the baseline. So, and you can see on the gel, the ladders have disappeared. So this is working in the right way, we think. Then we went ahead and made the mutations of the peptide. Of course, if you mutate the lysine residue that's getting ubiquitinated, you use a flat baseline. But if you mutate any of the PY motif residue, you have much reduced fluorescence polarization. And if you have something that blocks the active site cysteine, like an alkylating agent, you expect no fluorescence polarization. That's exactly what we see. And we can use that sort of inhibition to estimate its Z factor. The assay shows a Z factor of 0.77, which seems very good, and it's ready for high throughput screen. So right now, where we are going with this, we're now setting up high throughput assay, where we have this library of compounds from Purdue Drug Discovery facility. And now we are using this assay to look for basically compounds that block this signal. And I should tell you that some of these compounds will turn out to be because they block ubiquitination. So we'll also set up counter screen where you have just the HECT domain missing the WW domain. So to subtract those false positive. So now we can find compounds that will essentially are getting ubiquitinated due to their obligatory binding at the WW site. Okay, so that was like and we are still doing that. Unfortunately, I don't have any final compounds to share with you right now. So maybe in a future meeting, if I get the chance, I'll have something there to show you. But let's go to this HECT domain and how it's being HECT protein called E6AP and how it's being hijacked by a virus protein. So this is another HECT protein, E6AP. And normally, the virus protein is from HPV, and the protein is called E6. The E6 protein, when the virus infects human cells, cervical cells, the E6 protein of the HPV virus, HPV, now hijacks this ligase to ubiquitinate P53 and destroy it. Okay, so normally, E6AP on its own does not bind with any affinity P53. Okay, there may be some affinity, but people haven't detected it yet. And so E6 itself, again, nobody has shown it. We try to do this, but we cannot demonstrate or find any detectable binding. But like Ning mentioned maybe we go to high concentration and titrate it, maybe we'll see some binding, but we can say maybe at this approximately E6 on its own doesn't have any binding for P53, but E6 can form a complex with E6AP, and then that complex can bind to P53. And once P53 is bound, kind of like a molecular glue, proximity-induced ubiquitination of P53 happens, and it would be subsequently degraded by the proteasome. So this way, the virus E6 protein now uses a host E6 E3 ligase, HECT type of E3 ligase, to hijack and destroy a tumor suppressor, and that is believed to be one of the major causes of HPV-induced cervical cancer. There are other causes too. So of course, we know that this is a ubiquitin transfer reaction. It has to, the complex has to bind to ubiquitin charge E2, and ultimately that ubiquitin has to be transferred to the catalytic cysteine. So Kenny's interest and after Kenny graduated, my lab's interest is to capture individually these complexes in the bound E6 form to really understand the dynamics and the mechanism of, detailed mechanism of ubiquitin transfer. So a little bit of history where, I told you there was a lot of most interesting stuff already known before we entered the field. So I want to give credit to people who have already done the work. So this system was discovered in 1991 by John Hubres when he was at Harvard. And so basically it is E6AP is the system that he discovered was E6AP ends a protein and interacts with the virus E6. That was the original discovery in 1991. And E6AP, the full length of E6AP protein has 875 residues. And then Lian Huang, as I told you before, in 1999, solved the structure when she was in Nicola Pavletich's group with the HECT domain, which is, I think, from this 520 until the end, and with E2, UBCH7, the specific E2. And the structure beautifully at that point showed how a ligase can work like a ligase. Then fast forward in 2016, a structure appeared from a French group. It's a very important structure where they actually co-crystallize P53 with E6. And instead of, they're unable to capture the E6AP protein, but what they ended up doing in the structure is take this peptide segment that is called LXXLL motif, which is like a 10 residue peptide, and that binds to this pocket on E6, and that whole complex explains how E6AP would bind over here, and sort of the rest of the catalytic domain was absent, so you have to imagine how P53 will be ubiquitinated, but this captured essentially some essential feature of this ternary complex. Then relevant to our work, in a paper from a Japanese group, they actually painstakingly mapped the ubiquitination sites. I'll return to that in a few minutes using proteomics. And then more recently, in 2021, Kylie Walters' group published a paper that E6AP actually has a domain over here that binds to human RPN10, one of the subunits of the proteasome. And that raises an interesting question whether this whole hijacking system actually occurs when this ligase is bound to proteasome. Okay. All right. So with that knowledge, and we have sort of a combined background. Previously I showed you this cartoon, but now we sort of modify that cartoon, including this RPN10 binding to that RPN10 binding domain. So we are really interested in capturing the whole complex and hoping to understand how this would be recruited to proteasome. Okay, so one other thing we wanted to ask, which we talked to biologists, and they were really interested in that question, where E6 HPV comes in two flavors. One is called high risk, and one is low risk. Okay, and the high risk HPVs, for example, HPV 16 or HPV 18, and there are many other HPV strains unknown. Even if they infect you, they will not have any severe cancer-like phenotype. Okay, so they'll not cause cancer disease. They may have some benign growth, but not cancerous tissue. And so basically, it is believed that the E6 protein from low risk, they also bind in a similar way, but they somehow fail to induce ubiquitination of P53. Okay, And so I talked to John Hubris and all, who knows the field very well and in the biology side of things. And so one of the things we wanted to ask as we look at the structural data is, can we explain that how, in spite of having very similar structure, what is the difference between high risk and low risk HPV at the basis of that E6 interaction with the E6AP? And as I mentioned, we're also interested in knowing, does E6 target proteasome-bound E6AP? And then how does E6 evade ubiquitination despite being on the substrate interface? Right? People know in this field that many times when you study ubiquitination, the ligases themselves get polyubiquitinated, which could be an artifact of the in vitro assay. So, but in our assay, we would never see E6 getting ubiquitinated, although it's at the interface of the two proteins. So that's one of the questions we wanted to ask and get a structural answer to. And primarily, I think our structural goal was to really solve the full length, sort of look at the full length structure with E6 and P53 bound form. So this is called complex 1 because it is E6AP bound to E6 and P53. Complex 2 would be when it charged E2 bound. This is what we write them for convenience. And complex 3, we designated the one where this ubiquitin has been transferred to the catalytic cysteine. And today, I'm going to, these are work in progress. It turned out to be quite challenging. So I'm going to focus on what we have learned so far on complex 1. Okay, so first of all, one of the problems in this field was working with the full-length protein because it was very challenging to purify. And, so we dealt with that with a challenge, and basically what worked with us was we could co-express E6 and E6AP and then mix that with separately expressed P53, the cells, and lyse the cells together and then purify using his tag on some of these proteins, and then purify them further by size exclusion chromatography. And we could see that, for example, you have 1 or 2 fractions shown over here that contains what looks like a stoichiometric mixture of the 3 proteins. So with that, we thought that could be our complex 1 co-eluting. Then we went ahead and started doing structural work. But before we went there, we asked the question, is this complex enzymatically active? So basically, when you take this complex and add E1, E2, and ATP in a time-dependent way, you see the P53 level disappear and this higher molecular weight smear appear. And if you excise that high molecular weight species from the gel and submit for LCMS proteomics, and because we can now identify the peptide fragments that have lysine residue that have been modified with diglycine remnant of ubiquitin. So we could identify ubiquitinated peptide, ubiquitination sites on P53, and it is consistent with what the Japanese group, as I mentioned, observed before. So namely, this lysine 164 is a dominant site on P53, and I'll come back to that when I show you structure. Okay, so we also knew RPN10 can bind to the RPN10 binding domain from Kylie Walters' work. So we thought maybe we can add that to our system. First, we have to demonstrate that it actually binds to our complex. So we have HA-tagged RPN10, and we showed through an immuno precipitation experiment that HA-tagged RPN10 can, in fact, pull down the 3-protein complex. So we decided to include RPN10 in a complex sample, so with the hope that it may stabilize the complex, or it may give increased mass, which is always good for cryo-EM. Okay, so our cryo-EM workflow, we first collected 5,500 micrographs, and then did an initial 2D classifications, then liked one group, one class, and then we had an initial reconstruction of that, not at a great resolution, but we used this basic sort of shape as a template to look for more particles, and then from there we did one more ab initio reconstructions using the best class, and from there 3D refinement and post-processing to finally have a 3.54 angstrom model of the complex, and that's what the structure looks like. First of all, I should mention that this is again the full-length protein. We actually don't see the RPN10 binding domain. Neither do we see RPN10, but we know that it's present in our sample. So that part is probably unstructured, and so we cannot resolve in our cryo-EM model. So what is new in the structure? Of course, we have the HECT domain present, nearly identical structure as Land had solved before, and that sort of is the catalytic cysteine on the C lobe. And then we see an additional domain here that spans from 236 to 518. And previously, people thought that was unstructured. But in our structure, we see that is nicely a stack of helical helices. I'll show you that in a minute. So essentially, in terms of new elements of HECT E6AP, we have been able to resolve a portion of the N-terminal domain that now shows up as an ordered structure. We call it N-terminal ordered domain, or NOD. Okay. And so if you look at, and that's where the E6 is bound in the back, and then you have this P53 now over here. And so you can sort of see this is the lysine 164 which we mapped using mass spec experiment. You can see how the catalytic cysteine is so far away from that specific lysine. Okay? And which is a feature that, you know, people in the ubiquitin field know, Ning and others, that this is always when you crystallize or solve the cryo-EM structures, the HECT catalytic cysteine is always away from the substrate cysteine, but this is what we see in our structure as well. So let's look at the E6AP a little bit in more detail. So that's basically the structure that Land had solved before of the HECT domain. So what we have new is the rest of the proteins, which we divided into 4 regions, and this helical stack, and then there's this the backside helix. We call that backside helix because if you look at the complex, the E6 actually, this LX, I simply call it LXL motif. This is the motif that is known to actually engage with E6, and that's the one that was crystallized in the 2016 structure by the French group. Now we can see that helix is now projecting out some disordered loops from the rest of the N-terminal domain. And then if you look at the complex, in addition to that helix binding to E6AP, that red helix over here from the NOD also binds to the backside of E6, so we call that a backside helix for that reason. And then that's sort of shown over here. Maybe you can see that that's the E6 part that engages interaction with that helix, and that is the LX motif that projects out from NOD and gets into the binding pocket of E6. Okay, so now we, of course, there are some interesting residues, conserved residues that are now at the interface between the HECT domain and the NOD, and I'll not go into describing them. The main thing is that we are now testing the importance of this in a yeast-2 hybrid assay to see what these residues, how important is the interface in biological activity. Okay, so that's the complex. That is, now let's focus, in the interest of time, I'm just quickly take you through the 2 reasons of interest. One is this area where we call the backside binding area again. That's the backside helix. And so that's zoomed in over here. And this is mostly hydrophobic residues coming into contact. And there's a little patch over here, and there's this tyrosine and threonine from E6. They make contact on the NOD of E6AP. And when you looked at the sequence, these amino acid sequences from these 2 regions that we believe to be important for binding, they're kind of the same in all the E6 variants from different HPV strains. So there's no, we can't really say based on the structure, what is and what makes it a high risk versus what makes a low risk. And that sort of explains why the different E6 binds to E6AP, but it doesn't explain why they would not induce ubiquitination. Okay. So we suspect we have to look beyond this static picture, and we have to look at the dynamics of the system. And that's what we are doing with Alex Zelter and Rachel Clevitt at University of Washington. And so we set up this experiment of cross-linked mass spectrometry where you use this sort of lysine cross-linkers that will capture 2 lysines if they're proximal by 10 angstrom. And then so our idea is let's prepare different samples and add different components of this, the ubiquitination system to the sample and watch or characterize lysine residues that move within 10 angstrom of each other using this cross-linker. And so when we do this sort of experiment, this is just one series of experiments we have done. We are repeating it using different cross-linkers. And so basically what we see is that you expect many of these distances, although they're far away in static structures, sometimes more than 20 angstrom, but we know that the E2 shown in green has to move closer to the catalytic cysteine of the C lobe. So we see those long-range distances as expected. And we also see an interesting distance between this P53 and one lysine of this NOD. And what is that distance? It basically tells us that there is internal motion that brings P53 and perhaps that ubiquitin next to anchored on the catalytic cysteine, and they sort of use that as a platform for those things to move relative to each other. Okay, so and around this time when Kenny was doing this work, a paper was published in PLOS Pathogen where they, without the structure, they tried to ask whether they can map out interesting regions of E6AP in the context of the P53 ubiquitination. And what they found, and this is now I'm mapping that onto our structure, is that there is this blue helix, and of course this red helix is actually the LX motif that's known to be important. If you delete that or mutate that, there's no ubiquitination. And so they additionally found out this blue helix, and that's called B in our structure, and this green helix over here, that's from NOD, and these both are from the N-terminal order domain, and this is our backside binder, and this is the new sort of functionally important helix, and that helix is the one that we see establishing a cross-link to the lysine 164 that we have identified using P53 ubiquitination site mapping. So in other words, the reason if you delete this in East2 hybrid system, this whole system is non-functional, is because perhaps this is required in a transient state of ubiquitin transfer. With that, this is what we have so far, a complex one, and now I'm sort of docking a land structure onto it. We could imagine how the E2 charge E2 would bind there. And now Brenda Schulman's group and Kamadura et al. Have solved the structure of this state, and where they show that things have to move in order for that ubiquitin C terminus to be attached to the catalytic cysteine of the E2. So we sort of have a picture of how that would look. Now the next question would be, how would you imagine that C terminus of ubiquitin going all the way to the lysine of P53, okay? And that sort of is basically, it's a cartoon that we show how the arrows should move. I think the E6 has a lot to play with the changing the dynamics there, but we have no answer so far. That's what we are pursuing right now. I can stop here and answer any question you may have.